All right, it's great to be here. Really excited about it. As it was said, I'm Rob Sambursky. I'm the president of Verséa Ophthalmics, but I'm also a cornea specialist. And at Verséa, what we're really after right here is the opportunity to deliver innovative, decellularized biologic solutions. And these are really cost-effective targets. And what we're looking at is ocular surface disease and surgical wound healing. And so when I think about my past life, this is my fourth venture. I've always had a path to follow that had a lot of potential obstacles: clinical trial design, FDA approval, reimbursement, clinical adoption. And this time I'm coming at it from a totally different place. I'm actually sitting here right now with you, and I can share that we have access to very differentiated product line. This decellularized biologics open the door to very large market opportunity where there's a significant need. We already have the regulatory approvals, the RFDs, for all of our products. We have in place today large clinical trials, level 1 studies on wound healing, as well as ocular studies. We have reimbursement in place, and I'm proud to say that in our short life of 2.5 years, we're growing at 65% growth year over year, and we're up 26% already Q1 over Q4 of last year. And we're doing this because we have significant KOL support, both academic as well as partnerships with large private equity-backed clinics. And so when you think about decellularized basement membrane, has a lot of clinical applications. This can be for the classic persistent epithelial defects that you think of with severe dry eye. It can also be for neurotrophic keratitis. And thanks to John Hovannisian, we know it's grossly underdiagnosed. At the same time, there's opportunities to use it for microbial keratitis, for corneal dystrophy, the classic thermal or chemical injuries. Not only that, the graft is so unique, and because it's decellularized, we can do glueless, sutureless procedures. So that makes it much more efficient when you're doing pterygium surgery, conjunctival chalasis surgery. It opens the door to ocular surface reconstruction in different ways, even for Mohs surgery. And what I like the best is we have new data coming out on macular hole. We show significant improvement, not only in the closure of the hole, but improvement in vision. So if you take a decellularized basement membrane, you say, where does it fit? Traditionally, you had cryopreserved, and I think very highly of biotissue. And then you have on the other side, classic dehydrated, which I just call conventional dehydrated. But we're somewhere in the middle. We're actually decellularized basement membrane, which I believe opens the door to better handling. We get the advantage of being truly bidirectional. And what really differentiates us is that we have a more efficient healing process. And what I want to share with you is sort of the logic behind this. So when you have chorion, when you have actual stromal debris like fibroblasts or epithelial cells, you actually trigger inflammation. We don't have that. So in the end, we have no pro-inflammation at all. We have a process that's mechanical to create the decellularization, and we air dry. So we preserve the laminin and the fibronectin that produce the growth factors and cytokines, and we also produce a process that preserves the collagen and elastin that gives us a great matrix for cellular migration. And we know that when you look at the components of, so let's say, adhesion and proliferation, you compare it to cryopreserved and conventional dehydrated. If you just look at the bottom left, that picture is showing the first days of healing, and if pentraxin and high-chain hyaluronic acid were critical, there's no way we would have that level of proliferation, leading to more efficient healing. At the same time, if you look at it clinically, we can show in a paper that just was published, when you look at neurotrophic keratitis, you look at dry eye, at 1 day we had complete resolution of the defects, and at 1 week it still persisted. So ultimately, we have this decellularized technology. We're about to expand with Biovance into macular hole. We have Interfill, which is a flowable connective tissue that we see value for meibomian gland dysfunction, for Mohs surgery and oculoplastics procedures. And then we have Centiflex and Biovance that are ideal for glaucoma tube shunt as well as trabeculectomies. So that's my talk. I just want to say that we have a Series A that's being undergone right now. I have committed $7 million of 12. Anyone that's interested, please let me know.